recording.
You guys can hear us okay?
Yes.
Oh, fantastic.
So if you want to start sharing your screen.
Yes.
And again, thank you so much for accepting the invitation. And I'll introduce you very briefly. So we'll have, is that okay or should I?
No, that's perfect.
Okay. So it's my pleasure to introduce the company Abzu. They very kindly accepted our invitation to share a little bit what they do, and hopefully it will be helpful for many of the groups here at ICM and other people that are joining online. So we'll be hearing from Sam DeMarter, did his PhD in systems biology at the University of Oxford, and also Mira Machado, who got her PhD from the University of Copenhagen. And they'll be telling us about identifying interactions in omics data with their explainable AI system. So thanks again for joining us. And as you know, we'll be recording the talk. And yeah, over to you, Abzu. Thank you so much.
Hi, thank you so much. Really nice to be here, and it's great to see so many people in the room and online. Yes, we're here to talk a little bit about Abzu and the technology and the projects we're working on. Just to start with, we will, of course, make the talk more general than just clinical biomarkers, because clearly this tool we have here is a general data science tool that can be used on any sort of supervised machine learning problem. So yeah, really excited to be here. The way we'll structure the talk today is I will give you an introduction to Abzu and the team and a high-level fly-in on the technology. And then I'll describe a few use cases that we've been tackling with the technology in especially high-dimensional data sets. So, wide data sets with many variables that don't have many samples. So this is a very tricky problem that the technology is particularly good at. But we can hopefully together with you today explore how can we apply the technology on your research. That's what we're most interested in. We want to figure out how can we help you guys. So please ask questions on while we are talking in the chat, or even interrupt us if you want to. We really want to make this an open discussion. We are here to have a good scientific exchange, hopefully. So after that, the Mira will give us a run through a concrete example on a single cell data set on epilepsy, comparing human epileptic tissue to healthy tissue. Focusing on one excitatory neuron in particular to figure out what is the gene signature that potentially sort of describes the biggest difference between these two groups. And then we can try to learn something, whether this is causal or discorrelation. And then after Mira is done with that, I'll also show a few more cases to hopefully inspire you and make you think a little bit about how could this technology help in your research. So with that, I'll start the presentation here. So we are not that small anymore. We were started around 4 years ago by 7 founders. So this is pretty unusual for a startup company. The 7 founders are split between Copenhagen and Barcelona. So we have an office in each location. And we also recently opened an office in Basel, which is a bit closer to you guys. Maybe you want to come visit. And we are a team of very different backgrounds. We have physicists, mathematicians, we have bioinformaticians, economists. Yes, we like to tackle tough problems in the data science space, especially in the life sciences, where we really think the technology that was developed here by the team can add something very new and opens up a whole new opportunity in omics and other types of analysis. We are raising money as we are still sort of in gearing up to make a full commercial company, but we are also very strongly represented in the academic track. So the reason we're talking to you today is to, essentially communicate the technology, because it's for academic purposes and non-commercial purposes, the technology is free to use, and we encourage academics, and we even want to collaborate to further sort of show what can be done here. And then on the commercial track, we have, of course, also customers. We're very international, and we like to keep things sort of lightweight. But let's see, what is the technology here? So to set the scene, as many of you will know, AI and machine learning has often been plagued by the interpretability problem. So you can often predict quite accurately what a certain outcome will be on a given data set, but it's becoming, well, it is problematic, and it is becoming increasingly problematic to interpret these models because they're getting more and more complicated. So some people are building these very complicated models, and then they're trying to look into the model afterwards, sort of to try to understand what happened. And that can be quite challenging. So there are things like SHAP values and Lime values that can give you a hint as to which features are the most important in your prediction. But it's pretty tricky to understand the interactions between features, and you have to be pretty sort of advanced to be able to use some of these tools. So what we're hoping to bring to researchers with our technology is a transparent technology that was built with this in mind from the ground up, right? It's a totally new algorithm that has not been developed before. It's proprietary to Abzu. And you might call it a symbolic regression algorithm that finds mathematical expressions that fit your data. And Mira will be able to give you much more details on the actual algorithm later on. But what it then gives you are simple explanations as to what is happening in your data set. And this hopefully then also triggers creative ideas and an interaction with the algorithm and the researcher to further advance scientific discoveries. So on the left-hand side, we paint the picture of black box AI and how predictions are going through the roof in some sense, but explanations are lagging behind. And we're really hoping to entangle the researchers' minds in the research process by using powerful explainable AI methods. We don't have to compromise in performance anymore. We have algorithms now that can get the same performance or very, very close performance to black box methods while also keeping interpretability alive. So we have, in this talk, we'll focus on biomarker discovery. It's an interesting problem because it's, of course, clinically relevant. It is also a tough problem because often data sets that you use to find the biomarkers are very high dimensional. By that, I mean you have many, many measurements for a given sample. So the statistics are very challenging here. Generally, black box or any machine learning method tends to overfit on these problems. So that means it finds any sort of signal in the training data, but that very rarely then translates to the new set of data that comes in, or some sort of validation test set. And the reason that biomarkers are so interesting right now for companies as well, but also patients, of course, is 1 in 10 drugs only succeeds in the clinic. And that is after it has gone through all the preclinical work and all the toxicology. Even then, only 1 in 10 drugs succeeds in the clinic. And that's partly because we don't quite know who will actually benefit from the drug. There's certain genetic profiles, there's certain metabolisms, there's something in patients that makes them either respond or not respond to the drug. And if we can find out who these people are before we start the clinical trial or before we prescribe the medicine, we can help a lot of people, cut down on frustrating sort of regimes where the drug doesn't work, and companies will save money and everything will go a lot smoother. So there's a lot of motivation also from the FDA and other health bodies to get a handle on this. Because another problem is misdiagnosis and overdiagnosis. For example, in breast cancer, there's certain women who have some sort of sign of some sort of lesion or some sort of knot, and often women who are perfectly healthy and will never develop symptoms and metastasis still get treated and overtreated. So you're actually damaging a perfectly healthy person, and you are not helping them in any sense. So if we can sort of stratify and separate the women who will develop malign cancers from the women who are fine and will stay healthy in the future, of course, that is also a very important thing to do. So just for everyone to get on the same page, biomarkers are characteristics that are measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention. So that just means anything that tells you what's going on in an organism can be classified as a biomarker. For example, heart rate might be a biomarker. A certain levels of albumin in your blood could be a biomarker. There's even these digital biomarkers coming out now that it's anything that you can do with a smartwatch or with an app could also become a biomarker. So it's anything that helps you predict or separate patients into response, non-response, or who will develop a certain disease or problems like that. And there's different types. There's diagnostic, prognostic, predictive, and predisposition biomarkers. You can read what they are here. And this is where we're focusing today. So let me just go into the details a little bit. And just to bring it back home, so if you have on the left here a patient population, this is for a disease called NASH, for example, and you don't have a biomarker, then you see the top picture. You get like 3/4 of patients will be essentially diagnosed with a biopsy. So you actually have to physically go in and take a sample from the liver, and only 3/4 will actually be confirmed to have the disease. So you're doing a lot of biopsies and hurting a lot of people before you actually get a sort of a positive diagnosis. If you can find a blood biomarker or something that's less invasive and essentially predict who will has NASH before you go in and do the biopsy, you can really reduce the number of people you have to open up to do this diagnosis. So that's another example of biomarkers being important. So this is going into the technology on a high level already. This would be a COVID case where we're classifying severe from non-severe cases. This is a cohort from the Mass Gen Hospital in Boston, where people with breathing problems came in and were diagnosed with COVID. So this would be this group. So we have 300 COVID patients, and of those, around 100 people had severe COVID. So that means they died or they were intubated in the hospital. And the remainder was non-severe, so they were either getting oxygen or discharged from the hospital. And then we have something like over 1,400 proteins that were measured of each patient. And now we're trying to use this to say, what is the signature in these patients that explains the COVID severity? Why are these patients doing much worse than the others? So we take these measurements. It's essentially just a table of 1,400 columns and 300 rows. So that's the input, and then we have a column at the very end saying, is this a severe patient or a non-severe patient? And then our algorithm finds the signature that separates these groups optimally with as few features as possible, and we get very strong predictions compared to other machine learning methods. But keep in mind, we're also staying interpretable. So this is the model that was found. So from 1,400 features, we go down to 2 features, and it's only a linear model, so you could argue it's a very trivial thing. But imagine all the combinations that have been tried here and that would have to be tried if you also want to consider nonlinear interactions and more than 2 features. Because these things were tried by the Q-letters, but they haven't really contributed more signal. So what we're saying here is you don't need more than CLTA is a protein and IL-6 another protein in this relationship to essentially explain why certain people get severe or non-severe COVID. So when you then look at the biology is linked to EGFR signaling on the pathway level, and on the phenotype level, these features were linked to abnormality of the pleura and anemia. And if you look in the literature, these are commonly described as indicators of COVID severity. But we didn't have to read it up. We just looked at the data set and interpreted the features afterwards. So this is one example where the Q-letters can really sort of go through a large number of data points to help you figure out what's going on in the data set. Another example was a breast cancer study here, where we essentially have a TCGA data set. It's a cancer database by the Broad Institute. And we have around 700 breast cancer patients here. We have 2,000 features for every measure, for every sample. And the features are actually multiomic. So we have copy number variations, we have somatic mutations, gene expression, and protein expression. So you can imagine a very wide table again with these different data types and different features. And now we're trying to use these features to say, can we predict why certain women died and why certain women survived within a 4-year period of their breast cancer? And again, we find here a very simple model, only 2 features with an apolipoprotein and myocilin. These are, one gene is associated with obesity and the size of the adipocytes in the breast, and the other gene is associated with extracellular matrix and the adhesion of cells. So there could be something around, a mechanism here that captures a population of women that do very poorly, who are obese and some sort of prone to metastasis. This is highly speculative at this point, and we're still trying to qualify this story with academics. And again, the reason we also like to work with academics is because you guys have the right questions, you have the insight, you have the subject matter expertise. We're not saying, this algorithm can do it all. It's the interplay of these models together with the hypothesis and the biomedical literature that really brings us forward. So I wanted to highlight that the model is a nonlinear model. It's a Gaussian relationship. And that's indicated by this circle here in the decision boundary. So this would have not been found with any other machine learning method unless you would have included many more features potentially, right? But then it wouldn't have been as interpretable. And we should note that with this simple model, we can already start competing with random forests in terms of performance and other types of machine learning methods. I'm not sure, are there any questions by this time already, or are we good?
I have not seen any hands raised in the room.
Right. In case there are any questions, please feel free to.
So, yeah, actually, can you explain to us what is really the specificity of your approach? Because it's not totally crystal clear for me.
Yeah, and I think maybe that potentially I'll hand over to Mira to let her try to explain. But thanks, Claire. We'll try to address that. Just one more case here. We tried other data sets as well. But I think I'll just stop sharing now and Mira can take over to try to, maybe with the focus on trying to explain what's special about the technology.
Okay. And Mira, I'm going to move the microphone to you.
Yes.
Yes.
This is it. Okay, so I'll get into the details of how to use the technology, what the technology is about, and so on. So first of all, as Sam said before, we are in the supervised learning realm. So we cannot. This is where the QLATIS is handled. So if you imagine like a tabular data set, like this one, it's just like I didn't take it from anywhere. I just took it from my head. But this is how usually the data sets that we put on the QLATIS look like. You have a couple of independent variables. Here I represent them by this axis. And you have a response variable or an outcome, which I represent here by this y. And you can have all sorts of variables in here. For example, they can be float of the float type. They can be integers. They can be letters, which we call categoricals. And the whole idea of supervised learning is that you can somehow map your input variables to an output via a function. So this is basically what this little expression represents. And people use linear models, people use random forests, people use neural networks. And here we use symbolic regression. Like the QLATIS is a symbolic regression algorithm, which means that we don't make any sort of underlying assumptions of how this function should look like. When you have a linear model, for example, you make the assumption that it's going to be a sum, a linear combination of all your input features. And these are going to map to your outcome, and then you find the parameters, the weights. So with the QLATIS, you don't make any sort of underlying assumptions about that. It's actually going to go through the gigantic search space of the all different combinations between these features so that they can map this output. And I will illustrate that a little bit with this, this little image illustrates that. So this is our Q-lattice symbol. This symbolizes a relationship being made in the Q-lattice. And you have seen in Sam's presentation this sort of little graphs showing up, and they do represent mathematical expressions. So if you would represent, you have your feature x0 plus x1 equals y, this is how it would be represented as a graph. So, and this could be one possible combination of how you would get your input features, or some of them in this case, to map to your output y. And you could also have a multiplication, for example, and that's how you would be represented. Here you have x0, x1, they multiply, and they give you y. So it's basically like that. So every little, this is a graph, so every little graph, it's going to be a representation of a mathematical expression. And the Q-lattice, it's going to, I'll show you how it works very soon, but I can just tell you a little bit in the beginning. So you're going to load your data into it. And then it will know everything about, you will know your input variables, it will know your output, it will know what kind of problem you're trying to solve. I mean, as in regression or classification, I'll show that soon. And it's gonna first give all these different combinations of models, of little mathematical expressions. And then when it gives these combinations, then on your computer, you're gonna fit all these models. So you're going to see what is the prediction that they give, and you're going to assign weights and biases to them. So they will also have parameters. And then once that's done, we're going to see which models perform better than the others. And then in the next generation of models, you're going to have models that mutate from those, but you're also going to have other random ones that can compete with those. So it's this sort of mutation plus competition of new models that in this interplay, we can find very interesting relationships on our data set. So this is the importing the libraries. In this case, I'm just gonna import Find. This is our library that interacts with the Q-lattice. It can be easily installed with pip. And I'll just import pandas as well. And the data set that I'm going to be using is from this paper called Identification of Epilepsy-Associated Neuronal Subtypes and Gene Expression Underlying Epileptogenesis. That was an awful. And this is a UMAP representation of the different subtypes. And they are colored by different colors. And this is the same. And in this, because it's a lot of data, I'm just going to focus on this particular location here, which here is annotated by L23 Cooks 2 Frame 3. And this is the same UMAP. I'm sorry, they cannot be side by side. My computer is too small for that. But with the control and epilepsy patients, like painted by control and epilepsy patients. So this one had quite a difference in the gene expressions pertaining to the cells in it. So that's why I chose it, because it could be that it has more signal when we try to tell epileptic patients from non-epileptic people. And this is how the data set looks like. These are the cells, that's their sequence and who they came from, the sample they came from. These are the genes. And this is what we want to predict, whether they are control or epileptic patients. So true for control, false for epileptic patients. And I already took the liberty of splitting it in train and test, because since this is going to be like single cell data of different people, I didn't really want a person to be split in two. So before I loaded the data here, I decided to split them up based on their sample, which indicates which person those cells came from, so that a person wouldn't have cells on the train set and other cells on the test set. So, but this is just a little detail. And then in the end, the control that we have like around 5,000 cells that are control of the control type, and around 2,000 they are from epileptic people, epileptic patients. Oh. Yes, and this is the split. That's why this split is not usual, a little function that would do a train test split. I just took it from the data because I split it beforehand. And this is a little detail that I'm just gonna, this is a little detail that we can pass to the Q-lattice. And most often it has helped in the improvement of the models. Because here, as you can see, you have way more, around like double the size of true compared to false. So we pass the sample weight so that the underrepresented class, which in this case is false, gets a higher weight when we are training. So that means.
Question for you?
Yes, question.
No, sorry, I think it's very clear. Thank you so much. Actually, it really answered a lot of the question I was asking. I was just curious because we see only add and multiply. So do you have in your algorithm way to look at subtract or divide?
We do have more functions than that.
Okay. I was just curious if one factor would be, need to be subtracted in order to lead to the effect. I was just curious, like, but maybe you can explain that after you went through it. Sorry.
No, that they can combinations of multiplication and addition, and other functions will eventually show up. And yeah, if they are good, they will stay. If they are not good models, they will be thrown out. But yeah, I can show you. So once you install Fine, you can create this variable called the Q-lattice, which here I call QL. And then it's going to be a community Q-lattice. So that's exactly, it's non-commercial. So, but you can publish papers with it. You can have some fun with it. But it's for personal and non-commercial use. And here you can set a random seed to it so that you know that your next results are going to be reproducible, at least so that you don't lose completely control, because the Q-lattice is going to begin its run by sampling many models. And then to assure that it's going to be the same models that you're going to see, you can set a seed to it. And this is how you run the Q-lattice, and this is how you get your model. So we have this little function called auto run, which is a combination of this, which is like different steps of different things that the Q-lattice do. So first, the Q-lattice is going to sample models. Then we're going to fit those models to see which are good, which are not. Then we're going to tell the Q-lattice to update those models. Look, Q-lattice, this is what we like. And then the Q-lattice is going to give you new models. And that's how the iterative loop will go. So what you give the auto run function is your data, the data set, the train one. You have to say which column of this train set is the output, and in this case it's control. You have to tell it what kind of task is it, whether it's regression or classification. In this case it's classification, so we have to tell it that. And these are the sample weights that I told you guys before about, which make sure that the underrepresented class is not completely ignored, and the Q-lattice is not just going to jump on in this case, true, control equals true, since it has way more samples than the other. And I'm just going to let this run. It's just 10 epochs. It's going to take a little bit because it's quite a wide data set, but it's nice to see what's going to happen. And in this case, it's still loading all the possible inputs, because and in this case, it's like almost 2,000 inputs, but we'll get there. Just a little bit of patience. Yep. So this is the first top model that I found, and you're going to see it changing, because at each epoch, it's going to have a different model that is the best. And as you can see, it has been trying many models. In this case, it's now almost 5,000 models that it has tried. And as you can see, the top is changing a little bit. And you can also observe whether or not some of these features, some of these inputs are repeated on the next epoch. So for example, this MEG3 is still here, and it's the end and it's still here. And it was here not from the very beginning, but it showed up at some point. And of course, this is a data set that has a lot of inputs, and you can certainly You can certainly get way better models if you increase this number of epochs, but for the sake of time here, I'll just leave it at 10. And I think I'm gonna put this up here. And models, in this case, it's gonna be a list of 10 models. So this is just a list. I can quickly show you guys how they look like, and they're all gonna be different. And this one that we are seeing here, it's gonna be the one on the top of the list because it's the one that has the smallest BAC, which is a criterion that is gonna penalize models for being too complex if you don't have enough gain in your loss. So these are the different models that we have. And as you can see, this MEG3 I showed in the top models, but here is not, it's not on the other ones anymore. So these are a couple of examples. And as you can see, we have different functions that can show up in combination. So in this one, for example, has an inverse. This one has a squared function. So you can have like different sort of functions showing up depending on, of course, on the fitting of the model. And let's assign this name best model, and let's check some usual metrics. So you can call this plot function. And here you can see exactly the input names, so exactly which genes were chosen by this particular model. And here you can see the performance, like some usual classification metrics. AUC is one metric that we look at a lot. So in this case, a little bit above 0.9. And here some confusion matrices at a threshold of 0.5. And you can also see how this model looks mathematically. And sometimes it can be a little bit tricky to see this. For example, this is not a minus sign. This is, as we can see here, the name of this gene, but that's fine. This equation does know that. And so you can see here the addition of these 3 genes here multiplied by these other 2, which is exactly what we have up here. We have the addition of 3 genes, and then they are multiplied by these other 2. And this is quite a complicated model, but you can always, here in Autorun, you have a couple of different options. And one of the things that we usually do a lot to make sure that that we can still keep the interpretability is to assign what we call a max complexity. So you could have the same problem saying with max complexity 3, for example, that in this case, it would give you models of, it's gonna take a little bit to load, but it's gonna give you much simpler models. They're gonna usually have 2 features mapping to the output. And that can be quite interesting when you do this. One thing that we usually do is little by little increasing complexity to see that we are really understanding what kind of problem we're trying to solve. So yeah, it began with some fairly simple models. But now, yeah, so these are the usual, this is the max complexity of the models that we allow. And with these ones, you can, it's much easier to plot them, it's much easier to understand what's going on, and then with that you can slowly move up in complexity. That's one way, one possible workflow that you can get out of this. And with such a simple model, I can also show you, with the other one, it just would have been a little bit trickier to show a decision boundary. But with this one, I can show you a nice decision boundary like Sam has shown before. And in this case, it made this funny linear boundary here.
So I was curious to know indeed, how do you how stable is it? Like if you run it on the same data set, do you get variation? And how do you feel confident about knowing that your model is the best one? Like how do you proceed?
Yes, so the first one, there is a lot of variation, especially the more inputs that you have. So in this case here, the number of inputs is 1957. So there will be a lot of variation, especially for the very little number of epochs that I ran. So maybe if I run for a longer time, some things will start to settle in. So we have already seen MEG3 a couple of times. So maybe that is a gene that you would expect to show up more often. But there is a lot of variation, especially because it's quite a wide data set. And on the other question, how do you know that your model is the best? Well, we are dealing with wide data. So, that means you will have different stories that actually will perform more or less the same and can tell you you have different models that can tell you slightly different stories, but can still solve the problem for you. So one of the things that you can do is just turn on exploration mode and see what sort of features do you get out of this, what sort of different stories you get out of it, and which ones actually make sense, or which ones you would like to explore further. That's one way of going about that. But as I said, it's a wide data set. It's mathematically, you wouldn't have a single solution to it.
Cross-validation as well.
Yes, there are some. We have also experimented with cross-validation methods just to see how much signal the QLattice can get out of this data. And that is also one way that we can try to see what sort of models this cross-validation scheme showed up. But they will also be multiple stories, and they will also present things that probably people already knew from literature and things that are probably new that no one has seen yet, which could be good for further exploration.
And Claire, just to elaborate a little bit, it is one of the unique aspects of the QLattice is the fact that we do get these multiple models, right? And you raise a very good question, and it's something that we are addressing in different ways by using subject matter expertise, some sort of, confirming with orthogonal data sets or other sort of sources of information. But there is, in our opinion, not one perfect model. Because of the multi-collinearity of the data, especially in omics, you will have, when you find a feature that makes a good predictor, you usually find 5 to 10 others that could do the same job in some sense. So you can almost pick and choose from those if you, depending on your question and what might be easier for you to do or what makes sense in terms of your research. So we can certainly elaborate on that, and I hope that we could answer some of your questions, Claire.
No, I think it's very nice, and I think many people in the institute, some who couldn't attend this morning, would be really interested. So if you agree, we can share the videos also with the rest of the institute afterwards.
Definitely, thank you, Claire.
Thank you.
There's also a question online. I don't know if you want to answer now or at the end.
Yeah, from Sadiq. I don't know if I'm pronouncing it right. I'm sorry for butchering your name, maybe.
Wait, that's Mira here for the room.
Okay, so the QLattice is selecting the model using forwards and backwards propagation. We have a gradient descent algorithm that can do that. So I don't know if you guys are very familiar with neural networks, but in neural networks, of course you have an architecture that you have set already. In this case, that's not how it is. But it's basically the same principle. You forward propagate your data there, and then you calculate your weights and biases based on the gradients that you get from your model. And that's how you fit them. And then afterwards, you check the loss of each of those models. And then you also check how complex they are based on what sort of functions they have used. And that's how we calculate the BIC, Bayesian Information Criterion, which penalizes for complexity. So that's how the QLattice selects a model, and then it sorts a list. So in this case, I just decided to take the model with the lowest BIC out of this list of 10, but it might be that their losses are not so far off from each other, and you might even prefer another model or explore all of them and see if they have some recurring pattern or something like that that can tell you something about your data. So that, and the next thing, no, it does not use any sort of feature. No, it does. So it does one thing before training, which is it takes the mutual information between all your input features and the output, but of your training set. So it's what you pass to autorun. And then, but it does not eliminate any input with that. It just assigns a certain weight for, I think, the top 100 of them. Assigns different weights, and then it assigns the same weight for all the rest. It's a way for us to converge quicker to a good result. But a little bit more of an advanced user can change that. You can either turn it off, but not with autorun. With the functions that make autorun, you can turn it off, or you can pass on another type of feature. Not importance, I would say, but feature ranking. That's how I would call it. Because not always the feature that has the highest mutual information with the output will show up here. that's not necessarily going to happen. Yes, could you answer some counterfactual questions on this model? I'm not sure I understand this question.
Adik, maybe you can extend your question.
So maybe
Do
you
you
can
have
go
any
towards
ideas?
causality, and everything that we're claiming here is purely correlation, and we're saying if there is a causal relationship, we are likely to find it with these simple explanations because of Occam's razor and these tendencies that the simpler explanations tend to be the ones that are more generalizable. The counterfactual here, I'm also not sure exactly what you mean by that, but so one thing that we are exploring is, of course, you can look at the 2D response plot, if you can just show that. Okay.
Yes.
So what we're saying here is, right, you need to have, is zero epilepsy or healthy,
Zero is epilepsy, yeah.
So you need to have high MEG3, and you need to have a sort of low, pretty low RP11 to say that you have an epileptic cell or excitatory neuron. So that is the interpretation of the model. The counterfactual here, I guess you can decide, okay, what do I want to test? So one thing you can do with design queries. So if you had some sort of other hypothesis you wanted to test, you could put those features into the Q-lattice and ask it, hey, give me the best model using only these features. So then you would almost constrain it to say, okay, is there any signal in these features that I think are important or have read in the literature? You can also ask the question, I have this one feature and I want to know what its best partner is or its best interacting feature. So then you can say, okay, give me models that contain MEG3 and give me all the other features that play synergistically or orthogonally with MEG3.
Can do like this.
This is the
Yes.
This answer your question.
Make interventions in the model.
I mean, you could, yeah. You can play with it.
You could try playing with the model. I mean, this model that we are seeing right now, it has a what we call a query representation. So if you want to play with, you can play with it in different ways, right? So I have read about this TMLE method, which was something that people were trying to find causality, whether there's a causal relationship with some sort of binary variable in the model. And all that it needed for the models were the dot predict function of it. So if it's something about the prediction that the model does, it's going to output those in the classification setting as a probability score, and then you can try to try to find causal effects. For example, in this TMLE method. But if you're talking about the architecture of the model, you can access it in its own query string, which is something that you can actually pass to the Q-lattice. So you can take this function here, and then you can pass to the Q-lattice, and you would be able to generate models that are quite similar to this. And I would consider that as an intervention on the model, because you can modify it, but in a more like Q-lattice sort of way, that you don't just take this object here and intervene on this one, but use the Q-lattice to help you with your intervention.
Yeah.
And that's what also Sam was alluding to. You can take this expression and then you can say, I don't know if I want this exact gene. You can put it here. We can put exactly what's written here, this exact string. And then you could say, I don't know if I want this gene here. So just give me something, some other feature. And then it will do that. Of course, still showing up.
And while we're waiting for this, maybe Baptiste can ask his question, and then after that, Shaban can ask a question.
Yes, thank you very much for the presentation. I mean, you may have answered already the question, but I was wondering if you can force the model to include a specific feature that we know is of interest. Let's say, for example, the ApoE haplotype in Alzheimer's, or these sort of things. And somewhat related, can you include covariates or known confounders? For example, sometimes, very often, we know that age, sex, or site can have can cause a structure in the data that we actually want to remove in the prediction. So can you include this in your model?
Yes, for sure. As we can see here, this, well, I think it's the same. I think it ended up being the same. It's actually the same feature that it showed up here. Well, that's unfortunate.
So the top 10 feature models.
But yes, I will. But you, for example, with this query string, you can say, let's say you have on your data set the sex, age, and so on, and you want to include those, and then you can make a little query that will include these other variables. And yeah, I can.
Yes, but then I guess the way I understand it, so age and sex would be included in your prediction model, but what we're actually interested in is removing them from the prediction to make sure that the prediction remaining is not coming from those.
Yes, the easiest way of doing that is coming here to your data set and saying drop.
Yeah.
And then just telling the QLATIS not to run with
But
those.
I think I also have another answer, because I know what you mean as a bioinformatician, because of batch effects and also confounders, like you mentioned, sex, and so that the QLATIS does not handle. So this you would have to do with sort of normalization methods before you start modeling. One thing we have done before is, as Mira mentioned, use the QLATIS almost as a confounder detector, right? So you throw in all the metadata and you see which of my metadata is actually predictive of the outcome, and then you can learn, oh, gender is hugely important. Is this relevant to my research question, or do I want to regress this signal out in my pre-processing? So you can almost use it in that way as well, and it can be very quick to pick things up. But we do not handle these cofactors, I guess.
No, we do not handle them automatically, no. Well, it's something that you have to do it yourself.
Yeah.
So if you see that gender is a very predictive feature, but you do not want a gender bias on your models, then I would suggest you could stratify by gender or something like that. Separate your patients in a way that you have both representatives in train and test set.
Yeah, so good data science practice still applies in some sense, and we at this point don't handle that, unfortunately. But if it's something you really want to have, we could consider it, right, on our end. So please suggest things. Yeah. There have been two questions here now, and actually I will address both of these in the next slide. So maybe we can park them for now, unless Mira, or there's other questions on the notebook here.
No, I just wanted to just show the query string did work. Even though the top model is the one with the feature that we had found before, it still tried with other genes.
And this partly addresses the redundancy question. And likely these genes are correlated to each other. So RP11, CAP-DS, PS2, SLC38, these are likely correlated genes. Otherwise, they would not, well, yeah, there's a chance they are. And I will go into it in more detail in a second. Were there other questions on this notebook? Oh.
But I can still answer.
Yeah. I'll just share my screen here.
Any questions here? We have a question here.
Okay.
Okay, thank you for your presentation. I just would like to know whether there are some computational limitations in the number of regressors to be included in your model. Because for what I understand, you consider many kind of relationship between the variables. So I presume that it increases in a dramatic manner the number of possibilities. So I would like to know whether you have limitations in your model.
So at this point, we do not have that. It is always a practical consideration you should make to increase the training time. You know, if you can somehow do a variance filtering on your features. My experience is that around 2,000 features is a practical number when you want to interact with the QLATIS on the same day. if you want more features, then you would have to have it run overnight for a decent result. But what we do is, in order to handle that high number of features, is we calculate priors. So we use the mutual information of each gene or each feature as a way to tell the QLETers there's a good chance that this feature will be good in prediction because the mutual information is high. So we sort of tell the QLETers in advance what might be a good way to start. So that's how we deal with this high dimensional problem. But of course, as you mentioned rightly, the higher number of your features, the longer the training will take, and the longer you should run it as well, depending on how big your architecture is. If you only look for 2 feature models, you can be done within minutes. But if you look for 5 feature models, then you would have to learn overnight potentially. Yeah. So I hope that answers your question. I think it's great that these other 2 questions appeared in the chat. One is about unsupervised learning and can the QLETers find patterns in big omics data? Yes, it can, although we at this point don't call it unsupervised learning because we're still learning on a target variable, but it's finding subgroups in the data, and I will go into that. And then the other question was about redundant features, correlated features, and the question whether we're oversimplifying with 2 features. And I'll try to address that as well. But just maybe to start with that one, there seems to be some sort of almost like a data science or statistical law that when you have this wide data set and not so many samples, and by that I mean maybe 100 rows or 50 samples, you almost don't get away with more than 2 or 3 features without overfitting. And we've seen this on over 50, maybe over 100 data sets right now. So there's something about the statistics, some sort of phenomenon that as soon as you go higher in complexity, you just have no chance of generalizing on the test data. But it does not mean having 2 features in the model does not mean you only have 2 features to consider because of what we mentioned, the multicollinearity. So these 2 features actually represent potentially 20 other features that could also be predictive. But the essence of the signal is not more complicated than that. You have almost these variance groups, right? These features that essentially go up and down in very coherent and correlated ways. And you don't need more than that to predict. And as soon as you do need more, then you often start overfitting. But it depends, of course, also on the data set. Maybe we haven't quite seen the data sets that need more features. But it tends to happen that with bigger data sets, you can also add more features. And everyone knows that that's the case, of course, with overfitting becoming less of a problem. So actually, it's been on my mind, we should potentially quantify that and make almost like a guide as to, given your data set, how many features can I expect to have in my model without overfitting? But it seems to be something really generic that we're seeing over and over again. So that's something to ponder for you guys. All right, there's one more here, dynamic systems. We'll come to Joe's question in a second, I think. All right, this is a preprint we have been working on and we are trying to publish now. And I would refer you to that to answer not only biomarker questions, but generic questions on wide complex data sets and the QLETers. We'll be touching on the aspect of interpretability, feature combinations, multicollinearity, non-linear interactions. And we have touched up on each of these already, but just to give more details on it. We have seen that interactions are very important for prediction. So single features are often less powerful than smart combinations. That of course also makes sense. But you really have to find the right combinations, the right interactions. This is where the QLETers can help you. So this is a data set on Alzheimer's disease taken from the CSF of AD patients and healthy patients. And we did, well, not we, but the Matthias Mann group here in Copenhagen did a mass spec analysis. So we had something like 2,000 features, I think, in this data set. And again, we find very simple models. And at this point, we're actually taking the most well-known protein that is associated with AD, which is tau, or here MAPT. And we're linking it to age and another protein that seems to be adding a little signal here. And when you look at the response plot, that's what we're showing down here. So tau is plotted on the x-axis. As we go from left to right, the levels of tau are increasing. On the y-axis, we're showing whether the patient had AD, Alzheimer's disease, or not. So you can see this decision boundary, or this this response. As you go higher in tau, there seems to be a strong association with AD. The interesting part is if you vary the age and you go from young to older patients, the young patients develop AD with a higher threshold of tau versus the older patients. So there seems to be some sort of interplay between the age. When you're an older patient, it's lower MAPT or tau values that are going to put you in the positive class here, Alzheimer's disease. And that does, of course, make sense in some way, because there are, of course, many factors that contribute to AD, and the older you get, the less defense you have, in some sense, against the cognitive decline. But it's just a nice illustration of how you can use these simple models to start interpreting relationships. Another sort of feature here we want to highlight is the combinations of features. So in this data set, we have been classifying insulin response. So there was a group of women who were given, I think it was insulin, yeah. And then we looked at what was their sort of gene expression response. And we found a model with a couple of features. And what we want to highlight here is that each feature on its own separates the groups quite decently. You can see that by the distributions here. So if we look at this one, PDK4 separates the purples, which are the women who got the insulin, versus the negative, the zero class who didn't get the insulin. Separates it quite well, and the same for PHF23. But when you then look at the combination of them, they separate them much better, right? So this is just an example how the signal is adding up. It adds up to the model, separating the groups even better. And you don't need many features to do that. You just need to find the right features in the right combination. And that's what the QLabs can help you do. So we talk a lot about orthogonal features, features that are synergistic rather than correlated. This goes to the redundant features and the correlated features. So this is a model where we're predicting whether a patient had, I think it was liver cancer, hepatocellular carcinoma. And again, we only need two features. But if you look at the correlation heat map of the data set, of all features, so we plot all features against all features in a pairwise correlation heat map. The green blocks here show you the features that are highly correlated, and purple means uncorrelated. The model has found one feature from one block and another feature from another block, and that's all you need to predict almost perfectly who has liver cancer and who has no liver cancer. And this goes back to the idea of why do simple models do so well? Well, the signal, is there without having to add more. So, and that's what we're always balancing, is the simplicity versus the performance, right? Both because we want to keep it interpretable, but also because we want to generalize well on the next data set. We don't want to overfit. So that's really the consideration. And please don't be fooled by the simplicity. It's something that really is a powerful characteristic of a model when it can do the job with fewer features. Because actually one thing we didn't show, and it's always interesting to see, when we train the QLabs without penalizing for complexity, we can find all sorts of really interesting signal in the training data. And it grows like a tree, and get all sorts of interactions. And that's beautiful, but it does not generalize to the test set. And that's what the neural net would do, right? It would find all these different interesting combinations in the training data, but nothing stays true, or at least the majority disappears when you try to do the same on the test data. So, and again, just to bring that point back, so home, one way we like to think about it is in terms of maximum relevance, minimum redundancy. This is a concept that has been described before we came along, but it's something that we're tapping into. So you can think of the features in the model as the relevant features. And then you have features that are redundant and correlated. You don't need those in the model, but you could replace your relevant features with the correlated features if you wanted to. And then you have useless features out here. And the interesting thing, or the difference to what we're doing, is sometimes we can find two seemingly useless features, but in the right combination, they actually move into the relevant sphere, because together they actually add signal. But you would never have found them with a traditional statistical test, because there's no linear signal there. So I hope I'm not confusing you guys too much, but I hope this makes some sort of sense. But this is how we're thinking about this. And just to wrap it up here, we're showing how the QLATIS is being used in industry, but also with hospitals and academia for all sorts of problems. So we have already talked to Gautam about his research, and it's an interesting problem, but there needs to be some work done on the pre-processing of the data, because as you saw with Mira's example, the QLATIS sort of it takes in tabular data, where you have samples in the rows and you have features in the columns, and then you have a target variable, which could be a regression problem, or it could be a classification problem. And so the feature engineering and the data processing should not be underestimated. Not every research problem is immediately ready to be modeled with this tool. So we would encourage you to reach out to us if you need guidance, or think a bit more about how could you shape your data to feed it into the QLATIS. But I think that's it for now. And I think there was a question.
Yes, there was just a question.
You have a question from Joe on the chat, and then if there are any questions here or online.
Okay.
Yes, so dynamical systems. So we don't have anything automated to do any sort of time series analysis. So if you're talking about this stimuli as a function of time, we cannot just put it there and predict on how it's going to be like on the next time interval. So to make some sort of prediction on this data set, you would have to be pre-processed. So are there any time intervals you're interested in? Could you make different models from the beginning and a different model to the end of this stimuli? So you would have, for a dynamical system, we would have to get a little bit more creative on how we're going to use this and how we're going to make our predictions. And one idea is to separate it into different time intervals, for example, and then probably make different models. That could be one way forward. Or featureize your model in a way that you have the trend and the phase, the difference in peaks as well of your data set. Something like that could also be possible.
Yeah, my name is Tani, and I have another time-related question. Is it possible to kind of just take time out of it completely? So you have a data set that is time dependent, but you don't care about the time. So you kind of just want to basically normalize it to time. I guess that's, you just have a column of time that you maybe just, I don't know, what's the best way of doing that?
If you have a data set that's time independent, but you don't care about the column of time, drop it. Then you can just drop it. But of course, you should remember that there might be a causal relationship between one sample and the next, unless it's a time, it's something that at every second, it doesn't depend on the previous one, right? So there is that consideration to be taken into account. is every sample that you have at each time interval, are they dependent on the previous one or are they not? If they are all independent from each other, then fine, you can just drop your time column and you're probably going to have some interesting analysis that you can run on it without considering time. But if there is a causal relationship, if it depends, like the previous beep the current beep depends on the previous one and so on, then I would be very careful with just ignoring time.
What about Cox regression?
Oh yeah, survival models. Yes, so but we also had an example that one of our coworkers, he used the Q-lattice to make some sort of feature engineering as a feature engineering tool to put on a survival model called Cox regression. And he actually managed to make some interesting predictions with that. But he used the Q-lattice as a feature engineering tool in that case. So you wouldn't feed the model just a linear combination of the features. He fed the model maybe some feature multiplied by the other and so on.
But Tani, I think there might be some nuances here that would be good for us to understand. So if you could write it up and send it over, we can look at it if you wanted feedback on it.
Yeah.
Okay, cool. Yes, thank you. Thanks.
So we have 2 hours planned, but we are also happy to make it shorter today. So there were really a lot of interesting questions here.
Thank you so much, then. And yeah, if there are any other questions, you can write to me or to Abzu, and we'll also share the video that we recorded. And yeah, once again, thank you so much for accepting the invitation, and thank you to everyone who attended online and here. And yeah, see you soon.
Thank you so much.
Thank you.
Sam, thank you, Mira. Thank you, Elise. Thanks.
Thank you.